Welcome to this episode of Lectures with Liddington. Last episode, we talked about cellular respiration and what that process is. When I mentioned that there are 3 stages, that's kind of all we talked about, is the fact that there are 3 stages. You can go into aerobic or anaerobic respiration, but that there are those 3 stages. In this episode, we are going to dive deeper into those 3 stages and what's really going on. So let's remember with cellular respiration, we have 2 things going in and 2 things going out, right? If we write out our equation, we have glucose going in, C6H12O6 plus what gas? Yeah, that's right, oxygen. And then through cellular respiration, we convert those into CO2 and water. All right. Here is our friend glucose. Glucose starts off this whole process through glycolysis. Remember from last episode, that means to do what? Yeah, cut that glucose, right? Lyse is to cut, so we're cutting that glucose. So remember, up here, C6, right? It's a 6-carbon sugar. So the first process here in glycolysis is we take that glucose, which is 1, 2, 3, 4, 5, 6 carbons, and we lop it in half. We cut it into 2 3-carbon sugars. And that output, that 3-carbon output is called pyruvate. So we take our 6-carbon sugar and we end up with 2 3-carbon sugars, and those are called pyruvate. This is just our first step here, where we're sort of prepping for all the excitement that goes on afterwards. When we're done with glycolysis, we get a net of 2 ATPs out of it. So we produce a little bit of energy here, but not everything. This is just a starter step. So notice here in our diagram, there's nowhere that says anything about this oxygen being needed. And that's because this process is anaerobic. Remember, anaerobic means without oxygen. So glycolysis can occur regardless of whether there's oxygen present or not. When there's oxygen present, the next step becomes aerobic. And this is the Krebs cycle. So in an environment where oxygen is present, I will go into Krebs cycle. This diagram here on the side has a lot going on. But ultimately, for our purposes, what I want to get out of this is not to memorize all of the different carbon sugars that are filtered through this cycle, but just to understand that what we're doing is we take acetyl-CoA, which is sort of a version of pyruvate. Our pyruvate comes in here. So pyruvate comes out of glycolysis and pumps into this Krebs cycle in a slightly modified form. That's acetyl-CoA. And then it cycles through. We break things off, we put things on, we break things off, we put things on, and we do a bunch of different things with those molecules. But ultimately, what we get out of the Krebs cycle is carbon dioxide. So this is where I get my carbon dioxide, my product comes out of the Krebs cycle. Here in the Krebs cycle, we end up with a net of 2 ATPs per glucose molecule. So one glucose molecule will be broken into 2 pyruvates. So each one of these pyruvates will go into the Krebs cycle and pump out ATP. Then another one will come in and pump out another ATP. So I'll end up with 2 ATPs from that one glucose molecule that went through glycolysis. From here, we go into the electron transport chain, or ETC. Electron transport chain is a pretty complicated system. And in the electron transport chain, we have a very complex system of complexes. They are protein complexes that are found in this inner membrane. So out here, this is the outer membrane of the mitochondria, and in here, this is my inner membrane. Across this inner membrane, I have all these different complexes that are involved in shuffling things around. They shuffle electrons and they shuffle protons. And what this does is ultimately allows us to make that water, right? So water will come out of our electron transport chain. And then the other real important factor here is this guy. This is ATP synthase. And the job of ATP synthase, break it down, right? Synth is to make. So ATP synthase is making ATP. It will take phosphates and pop them on to those ADPs to make ATP. This is where I get the majority of my ATP molecules, anywhere from 28 to 34. It sort of depends on the situation. This is a little bit flexible. This is why in the last episode, when I said that you could get anywhere from 32 to 38 ATPs, this is why, is because here in the electron transport chains, things can change a little bit. But this process occurs across that inner membrane. So where all those cristae are, that's why there's so many cristae. That's why we have this folding. The more surface area we have to put ATP synthases, the more ATP we can synthesize. So we're really looking for an increase in surface area so that we can increase the amount of ATP production. So those are the stages of aerobic cellular respiration. Last episode, remember we talked about anaerobic, but here this is aerobic respiration. We go through glycolysis, the Krebs cycle, and the electron transport chain until ultimately we have our end product of cellular respiration. So let's take a pause here and do a little practice.